After studying this module, you shall be able to know the addiction as a disorder of altered cognition, learn the multi-stage cognitive process in addiction, identify the serious effects of addiction on cognitive abilities, evaluate the critical impact of specific drugs on cognition, analyze the development and impact of substance-induced disorders. In the previous modules, we have been acquainted with the concept of addiction, daily life substances used for addiction, and various approaches in terms of models that have been used to explain the addictive behavior. In the present module, we will focus specifically on the cognitive effects produced by addiction. The text will emphasize on the role of cognition in causing addiction and how in turn cognition is affected as a result of chronic addiction. Hence, the cause and effect vicious cycle of addiction is highlighted in the present text. Also, the learners will be able to differentiate the effect of drugs on separate cognitive faculties such as memory, executive functions, etc. The effect of exposure to drugs at different periods such as prenatal exposure, adolescent exposure, and geriatric impact is mentioned in the text. The potential ability of drug addiction that leads to other physical and psychological disorders is also attended in the text. The development of substance-induced disorders such as depression, anxiety, personality disorders, psychotic disorders have also been addressed. Cognition and addiction a link. Addiction since long has been viewed as a disorder exhibiting traits of compulsive behavior. The obsession with drug and resultant compulsive act of acquiring drug anyhow, abusing it to reduce craving, dealing with anxiety in case of absence of drug, all seems to mimic behavior of obsessive compulsive disorder. However, the recent work on addiction has developed a new perspective to view it as a pathology arising from altered cognition. Literature reveals that brain areas involved in process of addiction like hippocampus, amygdala, prefrontal cortex control major cognitive functions such as learning, memory, perception, etc., which reinforces the addiction cycle. The continued use of addictive substances changes the normal of the functioning of these regions and produces cognitive transitions that maintain the addictive cycle. Addiction has also been viewed as incorrect SR learning, where the individual may develop pathological connections between the drug and environmental conditions that may promote addictive behavior. The chronic use of drug then may produce cognitive errors or deficits which may obstruct the ability of the individual to achieve complete abstinence. Addiction has been viewed as a multi-stage process. It occurs in two stages where at the first stage, individuals' social drinking turns into pathological drinking and alters the circuitry reward systems of the brain. Such changes produce pleasurable effects in brain and encourage the individual to engage in satisfying pleasurable impulsive activities such as eating or sex. At the second stage, the addictive process begins to crystallize and individual starts depicting clinically significant pathological symptoms such as withdrawal signs, inability to achieve abstinence, greater chances of relapse, cognitive deficits such as impaired judgment, attentional biases, etc. Therefore, the initial stage marks the formation of pathological drug-stimulus association that reinforces the drug use, and the later stage introduces changes in cognitive processes that support the cognitive cycle and inhibit process of abstinence. Hence, control at the very first stage may have better prognosis than attempts of correction at the second stage. Cognitive effects of addiction. First, formation of stimulus-drug association. It has been widely observed that chances of relapse are extremely high for substance abuse and addiction as a result of strong maladaptive connections made between substance and the environment. These environmental cues trigger the mental associations and serve as strong precursors to the addictive behavior. Encounter with situational cues such as specific place of drinking or company or glass creates intense craving and urges the individual to engage and re-engage in the process of addiction. Exposure to such prompts introduces strong physiological response in the individual and at the same time produces changes in the brain activities controlling executive functions. This phenomena is called conditioned place preference and has been well depicted in use of various drugs such as nicotine, cocaine, amphetamine, etc. The research on various laboratory animals such as rats has also confirmed the phenomena where the use of drug in one compartment of the cage elicits a physiological response of craving when brought back to the same cage. The association exhibits the process of classical conditioning where the cage or place acts as conditioned stimulus and acquires the capacity to biologically elicit the response of craving even in the absence of unconditioned stimulus, that is drug or alcohol. Such maladaptive learning prevents relapse and restarts the chain of addiction. As a result, complete abstinence to be achieved is a challenge. Hence, it also warrants the intervention programs to include clear instructions to the significant others of the addict to avoid encounter with any of such environmental cues, like for instance, in case of alcohol addiction after abstinence, the family of the patient is instructed to remove the glasses of alcohol and not to use any fragrance which may have alcohol in it, as it may act as a trigger to the strong cravings and initiate the cycle of addiction. Second, persistence of stimulus-drug association. The persistence of this stimulus-drug association has been found to underlie the process of addiction. Also, the chronic use of various drugs have been observed to influence the association and prevent relapse. Researchers depict that addictive substances work on the communication pathways or synaptic connections between neurons to form and maintain the stimulus-drug association. Studies have further suggested that proteins that are involved in these synaptic connections are also involved in drug-seeking behavior, and hence as a result the addiction cycle is continued, which further prevents the relapse. The drug works in two ways with the synaptic connections, that is, strengthening of neural connections by long-term potentiation and inhibition of neural connection called long-term depression. Each of the drug exhibits either excitation or inhibition of the synaptic pathways to maintain the addiction cycle. Third, impact of drug abuse or executive functions. Considerable amount of literature has been available to document the harsh effects of substance abuse on cognitive functioning. One of the most common symptoms that emerge as a result of alcohol addiction is Wernicke-Korsakoff syndrome. The syndrome primarily manifests with alterations in memory function and poor emotional control, however preserving the intellectual functioning. With its debilitating effects on memory, it has also been included in DSM-4 as alcohol-induced dementia. In case of other substances, commonly decline in attention and memory have been found. In case of depressants, fall in psychomotor speed, temporal ordering and recent memory are generally viewed. Various stimulants have been observed to produce deficits in cognitive flexibility, working memory and attention. Depressants produce inhibition in central nervous system and lead to drowsiness, increased inhibition, slowness, sleep, etc. They also tend to slow the brain process, alter perception, produce attention and memory deficits, fatigue, heart rate, etc. On the other hand, abuse of hallucination drugs such as LSD, PCP, etc. lead to perceptual distortions, emotional irregularities, cognitive deficits, and physical damage. Another substance addiction observed most commonly in children and addicts is the use of inhalants such as nail polish, glue, eraser, etc. Chief signs of inhalant abuse include euphoria, dizziness, slurred speech, poor psychomotor coordination, hallucinations, delusions, etc. Its chronic use may result in poor inhibitory control, confusion, drowsiness, nausea, etc. In case of behavioral addictions such as gambling, food, sex, internet, persistent preoccupation with mental content, and performance of compulsive behavior emerge as major cognitive behavioral defects. Poor impulse control, feeling of helplessness, impaired judgment, biases in information processing emerge as central cognitive features in initiating and maintaining the addictive behavior. Fourth, cognitive effects produced by specific substances. There are various substances such as stimulants, depressants, hallucinogens that lead to different effects on cognitive and social well-being of the individual. Each of these substances may initially improve the cognitive functioning and emotional well-being, but with its chronic use, it may reduce the quality and well-being of cognitive and emotional functions. Some of the important substances used regularly have been listed as follows. First, nicotine. Nicotine dependence syndrome is observed widely and majorly amongst adolescents. It is found in items such as cigarettes, chewing tobacco, and cigars. The cognitive effects of chronic addiction to nicotine involves strong craving for substance, irritability, frustration or anger, anxiety, difficulty concentrating, poor implicit memory, restlessness, decreased heart rate, and increased appetite or weight gain. The withdrawal symptoms include headache, decreased metabolic rates, tremors, impairment of performance, etc. Second, cocaine. Widely used as a stimulant, chronic addiction of the same involves serious cognitive deficits. Memory deficits have been observed in various psychological studies. Poor immediate verbal recall, recognition, and diminished working memory capacity emerge as significant memory impairments. Studies have implicated role of cocaine in inhibiting the generation of new hippocampus cells and that reduce the functioning of working memory. Deficiencies in learning and memory have also been in cocaine patients even after remission. Amphetamine. The psychostimulant drug has been observed to enhance cognition in hyperactivity disorder and other neuropsychiatric disorders. It has also been viewed to improve cognitive abilities in healthy controlled subjects. However, its chronic use has been associated with significant impairments in cognitive functioning. Poor performance on various cognitive tests have been viewed as a result of cocaine abuse. Impairments in memory have emerged as the most significant cognitive deficit, along with the reduced attention span, which could be attributed to the reduced neurogenesis of the brain and disruption of blood-brain barrier. Fourth, ecstasy. The widely available illegal drug in the category of hallucinogens is not for clinical use. The chronic use of this psychotropic drug is viewed to be selective and persistent damage to central serotonergic nerve terminals impacting the areas of memory and learning. This reduced activity in serotonergic and dopaminergic is believed to cause serious alteration in perception, hallucinations, mood and memory impairments. Decreased working efficiency, reduced appetite, sleep disturbances and diminished well-being emerge as important psychiatric deficits. Poor performance on tasks of contextual memory, spatial memory, working memory, set shifting and cognitive flexibility is observed as a result of persistent abuse of the substance. Fifth, opioids. The version of opiates such as heroin or morphine have depicted to result major cognitive decline. The illegal drug is observed to produce attention deficits and memory impairments. The effects manifest in reducing pain sensitivity and producing significant impairments in learning and memory. The drug works by altering the functioning of hippocampus and the prefrontal cortex. Also, it inhibits the neurogenesis, contributing to serious cognitive deficits. Sixth, barbiturates. These medicines were initially used by doctors to produce relaxation and improve sleep disturbances. But with the heightened use of the drug, they realized that it produces effects similar as coma or death. Since then, the use of drug was restrained and was replaced by benzodiazepine. The drug works by enhancing the functioning of inhibitory neurotransmitter GABA, which then relaxes the brain and calms the body. The symptoms of addiction include tolerance, impaired judgment, confusion dependence, psychosis, blurred vision, slurry speech, hypotension, poor psychomotor coordination, irritability, depressive mood, respiratory problems, etc. The long-term effects of the drug include interpersonal relationship problems, seizures, attention and memory deficits, coma, liver disorders, heart problem, and death as well. The withdrawal symptoms include delirium, convulsions, irritation, anxiety, sleep disturbances, hallucinations, general weakness, etc. Prenatal exposure to substance abuse. Prenatal period is believed to be the foundation stone of human life. The development of brain during this period is a continuous process and is extremely vulnerable to any type of injury, trauma, or harsh exposure such as alcohol or drugs. This vulnerability is not restricted only to the prenatal period but also extends throughout adolescence. Such exposures may seriously affect the normal development of the brain and may predispose the child to various physical and psychological abnormalities. Prenatal exposure to alcohol may produce grave effects such as mental retardation or greater susceptibility to other substance addictions as included in fetal alcohol spectrum disorders. This spectrum of disorder is viewed to be the most common cause of mental retardation and other psychiatric abnormalities in America and other developed nations as well. There is no dearth of literature to confirm deleterious effects of substance abuse in the prenatal period. In one research, the children of alcohol and drug abused mothers were compared with normal healthy controls. The results revealed significant deficits in children of exposed mothers in the area of language, articulation, impulsivity, and visual attention span. However, the two groups did not vary on the parameters of intelligence, manual dexterity, or sustained attention. Another analogous study validated memory and attention deficits in children with mothers who were exposed to marijuana or alcohol during pregnancy in comparison to children of non-drug exposed mothers. Prenatal exposure to stimulant drug called methamphetamine was highly correlated with cognitive deficits and altered brain formations. The children with prenatal exposure to such drugs were found to exhibit memory deficits in terms of long-term memory, spatial memory, and visual motor coordination. Changes were observed in the brain structure of parietal cortex and frontal cortex of young toddlers whose mothers were exposed with psychostimulants. Similar effects were also viewed in laboratory experiments on pregnant rats. The expecting mothers were exposed to methamphetamine and the babies were found to be deficient in spatial memory. The effects of prenatal nicotine exposure is as debilitating as any other drug but exhibits greater concern due to high prevalence of tobacco consumption amongst women. The prenatal effect of tobacco has been well correlated in children with changes in hippocampal and parahippocampal areas. The studies also confirmed reduced cortical thickness and structural changes of white cortical matter in children with prenatal exposure to tobacco in comparison with healthy controls. The prenatal exposure to tobacco has been observed to lead to lower grades of intelligence and attention deficit hyperactivity symptoms in exposed children in comparison to non-exposed children. The results of various studies have confirmed a difference of as much as 12 points in full-scale IQ of prenatally exposed children and non-exposed children. The probability of acquiring ADHD was 3 times higher in children with prenatal exposure to addictive substances. Similar results of harsh nicotine exposure have been viewed in rats where it was related with reduced memory, related neural activity and changes in hippocampal area with both male-female rat babies. However, these deficits seem to persist only in male rats in later adulthood. Nevertheless, the cognitive deficits as a result of prenatal substance abuse seem to be grave in both humans and animals. The most serious effect that emerges is that it predisposes the fetus to later life addiction. However, it may not ascertain any specific type of addiction. This then may follow in further generations and may create a vicious cycle of addiction. Hence, it seriously warrants expectant mothers to avoid any kind of substance abuse in order to protect the fetus from prenatal damage and break the vicious cycle of addiction. 6th, adolescent exposure to addictive substances. Adolescence is the most delicate period of acquisition of any type of addiction. Majorly attributed to peer pressure, family exposure, societal and cultural norms, the process of addiction begins at adolescence and continues till later life. It has been observed to have debilitating effects on cognitive abilities such as learning, memory, attention. The most common substance of addiction during adolescence is nicotine, which is observed to have serious effects on cognition. Various studies in both humans and animals have confirmed the same. One study reported poor performance of adolescent smokers on tests of working and auditory memory, verbal comprehension and mental mathematics. However, most of the deficits seem to fade with termination of smoking, but deficits of working memory and oral mathematics seem to prevail till later adulthood. Even in rats, adolescent exposure to nicotine has been well correlated with poor visuomotor spatial abilities, impulsivity, attention deficits and alterations in the medial prefrontal cortex. Various evidences have confirmed deficits in learning and memory of rats treated with nicotine. Some researchers have also demonstrated initial cognitive enhancement with use of nicotine, but later major cognitive decline with its abuse and tolerance developed by the body. Nicotine abuse has also been found to lead to various other disorders in later adulthood, such as depression or stress-related disorders, which act as further reinforcers to the process of nicotine addiction. Abuse of various other substances such as stimulants, hallucinogens have also been found to lead to various cognitive deficits of memory, attention and learning. Thus, exposure to each substance in adolescence should be controlled and underlying factors should be appropriately treated. 7th, substance abuse leading to other disorders. Abuse of drugs may cause serious cognitive deficits, and at the same time people with mental illness tend to abuse drugs at the most. As a result, drug abuse becomes a cause and effect of acquisition and maintenance of mental disorders. Smoking and schizophrenia have been observed to exhibit high comorbidity. The symptoms of schizophrenia cause serious cognitive defects in the areas of visuomotor coordination, intelligence, impulsivity, perceptual distortions, etc., and its comorbidity with nicotine causes further cognitive decline. Nicotine abuse has been observed to increase the occurrence of schizophrenia, specifically in patients who are predisposed to develop the disorder. In some other cases, It has also been found that patients of schizophrenia tend to smoke more as a result of side effect of antipsychotic medications. Further, in certain cases, they use smoking to enhance their capacity to deal with tough symptoms of psychotic pathology. Drug abuse in early childhood or with prenatal exposure has also been associated with attention deficit hyperactivity disorder. As in case of schizophrenia, nicotine is also at times used to reverse the symptoms of hyperactivity, which in turn may also act as catalyst to the maintenance of the disorder. Besides nicotine, other psychoactive substances such as stimulants, psychotropic drugs, hallucinogens have also been found to alleviate ADHD symptoms. During intoxication, drug abuse is also associated with precipitation of various substance-induced disorders such as delirium, anxiety, amnesia, depression, etc. Learning disability, exhibition problems in the areas of reading, writing have been found to occur as a result of drug abuse. Problems in the area of problem solving, decision making, following instructions are also manifestations of substance abuse. Personality disorders such as paranoid, narcissistic, borderline disorders have also been closely associated as both cause and effect of the drug abuse. Depression and bipolar disorder and post-traumatic stress disorder may precipitate as residual effects after abstinence. In case of geriatric population, higher sensitivity to toxic effect of the drugs on the brain is viewed. Also, at the same time, they tend to be more vulnerable to cognitive impairments such as dementia or amnesia as a result of drug abuse. Thus, the use of drugs not only causes physical harm but very serious psychological damage across periods of development. Be it prenatal period, adult life or old age, substance abuse has been well associated with cognitive decline. As the acquisition, the treatment of addictive behavior is equally complex and multifarious. It has to target the period of acquisition, type of drug abused, reinforcing factors of addiction, dealing with withdrawal symptoms, comorbidity of other pathologies, etc. Hence, each intervention module has to be completely customized in order to be effective. Summary. First, addiction is viewed as pathology arising from altered cognition. Second, specific brain areas involved in process of addiction like hippocampus, amygdala, prefrontal cortex control major cognitive functions such as learning, memory, perception, etc. Third, the addiction cycle focuses on changes in cognitive processes as a result of substance abuse and parallelly maintenance of addictive behavior as a consequence of changes in the cognitive abilities. Fourth, addiction can also be seen as maladaptive stimulus-response association where the individual may develop pathological connections between the drug and environmental conditions that may promote addictive behavior. Fifth, addiction occurs in two stages. At the initial stage, the stimulus-drug association is formed, and at the later stage, changes in the cognitive processes that facilitate addictive behaviors are introduced. Sixth, the second stage inhibits the process of withdrawal and reinforces the addictive cycle. Therefore, control at the first stage may have better prognosis than attempts of correction at the second stage. Seventh, maladaptive stimulus-drug associations such as environmental cues serve as strong precursors to addictive behavior. Eighth, the persistence of stimulus-drug association has also been attributed to synaptic plasticity exhibited by various drugs. Eighth, drug abuse has been related with serious cognitive impairments such as attention, memory deficits, difficulties in problem solving, decision making, impaired judgment, mood instability, impulsivity, etc. Ninth, substance abuse during prenatal period produces deleterious effects such as mental retardation, ADHD, learning disability in the developing brain of the fetus. 10th, Adolescent exposure to drug is primarily attributed to peer pressure, family exposure, societal and cultural norms. It produces debilitating effects on cognitive abilities such as learning, memory, attention, judgment, etc. 11th, chronic drug abuse has been found to lead to various other psychopathologies such as schizophrenia, personality disorders, ADHD, learning disabilities, mood disorders, etc.